everybody, this is Heather Collins from Young and Savvy Genealogists, and today I'm starting a new how-to series on the basics of genetic genealogy. And so since this is going to be a series, you want to make sure you not only check back to see as we cover new topics, but also if you have any requests for topics that you'd like us to cover within this realm, definitely be sure to let us know and we'll be sure to include it. So to get started, we're going to talk about some basic human anatomy. Many of you will remember from middle school or high school learning about the human cell. And the human cell has all kinds of cool things inside of it, the endoplasmic reticulum and things like that, things that are responsible for keeping us alive on a microscopic level. Inside of the human cell is the nucleus, which is essentially the brain of the cell. And inside of the nucleus are the chromosomes. Chromosomes are basically the building blocks of all life here on planet Earth. And the difference between living organisms essentially is the number of chromosomes that they have. The difference between us and other animals is they either have more or less chromosomes than we do. Humans, as a species, have 23 chromosomes. 22 of them are essentially what form up the autosomal DNA test. And then the 23rd chromosome are the gender-based chromosomes. And so a son will receive a Y chromosome from his father and an X from his mother. And a daughter will receive 2 X chromosomes, one from father and one from mother. That's what makes up the 23rd chromosome, but regardless of the type of chromosome that it is, they're made of DNA. And so you see the DNA strand here, and we're basically going to be talking about the basic bits and pieces of the DNA double helix and how it relates to the basic concepts in genealogy. The first thing that you need to understand is that our DNA is 99% identical to every human who is alive right now or who has ever lived. Because our DNA is what's responsible for making us human, there's not going to be a lot of variation throughout most of it. It needs to be the same between all of us in order to define us as being a human being. But if our DNA were 100% identical, children would be exact replicas of their parents. There's no way to tell mine from my mother's or from my grandmother's or from my great-grandmother's. It's all exactly the same. And so the fact that there is variation, there is change that happens here between generations is exactly what makes it usable to us. These little rods between the double helix are responsible for how these changes happen and where and when they happen. And these little rods are called alleles. Each individual color here is an allele, and so they form together to form a pair of alleles. And pay attention to these colors because they correspond with these nucleobases over here. You have cytosine, you have guanine, you have adenine, and you have thymine. And they're represented with the letters that they start with, C, G, A, and T. They will only form in certain combinations. And since our material as humans is 99% identical, these are going to form in identical pairs for most of the time. But there is a 1% of time where they are going to change. And that is part of what we want to pay attention to in genetic genealogy as part of our first concept that we're going to talk about. Every time that these pair of alleles changes from what they were before, that's called a SNP. The reason that those are important is because since 99% of our material is exactly identical, anything that is different, anything that has changed is what is visible to us. These mutations are absolutely essential for what we're trying to do because it's when we can see and track the changes between generations. It's something that we can track that has taken place over time through various generations as they are transferred onto our new chromosome. So it happened between my great-grandparents and their children. It happened between those children and who would become my grandparents. It happened between my grandparents and their children, and then my parents and me. And so these genetic variations are something that have happened over and over again, and they are traceable and trackable on my chromosomes. But there are a couple different things that we have to be able to do in order to see those, to measure those, and to track those. And so the SNPs are the changes. And in order to track them, we have to measure them. And we measure the distance between SNPs in a unit called a centimorgan. You'll see that represented with a lowercase c and a capital M. You can almost think about it in centimeters, but that won't help you very much because you can't think about it in terms of geometric measurements like a standard centimeter. You can't think about it like that because SNPs happen on DNA strands, which make up chromosomes, and some chromosomes are more dense with SNPs than others. And so let's say you have, a 5 centimeter segment of a DNA strand. Just say that you have that. That same strand can have 3,000 SNPs on it. It can have 50 SNPs on it. It can have 1,100 SNPs on it. There's not, the SNPs don't happen at a standard placement. It's not, okay, every 2 centimeters there's going to be a SNP. That's not the way it happens. It happens completely randomly, and so it's not something that you can measure concretely in centimeters or inches. So we measure it in something called a centimorgan. As genetic genealogists, what we are trying to do, we're trying to find a measured matching segment of these SNPs to another person. That is our goal. When we're trying to match ourselves to another person, that's what we're basing it on. We're basing it on a length of SNPs that is of enough of a length and has a high enough SNP count that it registers as a match to us. So that's part of the half of how we are making a match. The other half is based on a process called phasing. And so we've pretty much established the measurements that we're doing. On top of these measurements, we have to isolate what is happening based on our mother's DNA and our father's DNA. Being able to sort which of the DNA came from mom and which of it came from dad is what's going to help us place our DNA into a generational context, not only between grandmother and great-grandmother and great-great-grandmother, but on which side of the family. And the easiest way to go about obtaining this is to have another person in your family tested. It needs to be a close relationship to you, and so whether it's a close cousin or hopefully one of your parents, a sibling, anything is going to help you to really sort that out. And those two processes work together, forming a biological map of your relationships. Looking at this image of this double helix here, this is a really simplified, not true to life version of what is happening here. Imagine that this red piece right here, imagine again that this is a zipper and it's zipped up the middle, so half of these little rods, these green rod segments came from mom and half of them came from dad, and they're divided down the middle. Imagine that the red half came from dad. Imagine that the blue half came from mom. So when you are talking about matching to another person, if you're talking about anyone who is not your sibling, you're talking about only matching half of this helix. So if it's a third cousin from my mom's side, that person is only going to match me on this blue part, this blue segment, and that match is going to be based on these alleles, this half of these green rods. These are going to match for a certain duration of this segment. We are going to have the same exact letters here. What happens when you share a parent is that these are going to be identical on both sides for a certain duration. And seeing how my segments compare to my sibling segments, they're going to help me to begin to phase these to see what comes from dad and what comes from mom. A sibling is the only person I'm going to match on both sides of the double helix to, because they are the only people that I have that I share parents with. And the more closely related I am to somebody, I'm going to have longer identical segments, and I'm going to match on both sides of the double helix. For most of the matches that you're going to be working with, that is not going to be true. For distant cousins, you're only going to match on half. And so the challenge is how to identify these lengths of segments and how to isolate them so that we can identify and establish who our shared common ancestor is. The higher our SNP count is and the higher our centimorgans are to our matches, the more related we are going to be to our match. That concludes part 1, and so stay tuned for part 2. We're going to be talking more about chromosome mapping and how to go about making these matches. If you have suggestions for other topics that you want us to cover in our genetic genealogy series, be sure to let us know in comments. Also, be sure to link, comment, subscribe, and share. And check out our blog at youngandsavvygenealogists.blogspot.com.